You want me to attempt to, I guess, go through the text and see if there's anything that we need to talk about. So chapter 2 covers chemical principles. At least that's the title of it. And so it starts out by talking about atoms, and then it talks about different types of bonds between atoms. The study of chemistry is basically the study of the interactions of atoms. Once you start getting into really how atoms work, you're starting to get into more physics than chemistry. And biochemistry is the study of the chemical reactions that go on inside the body. And in covering the basics of biochemistry, what they've done is talk about the different types of macromolecules. And so these are things like the carbohydrates, the proteins, the lipids, and the nucleic acids. So these things are all relevant to microbiology. They're relevant in two different ways. One of them, really more important to us as we're going to be learning microbiology in this class than the other. The one way is basically that to understand how life works, or to understand how microorganisms work, you have to understand how the molecules work, or at least if you want to understand it at that level. And to a degree we're going to be talking about that. We are going to talk about the genetics of microorganisms, and when we do that, we'll be inevitably referring to nucleic acid mutations and stuff like that. And we're also going to talk about the ability of organisms to replicate, and really, for example, the temperature range over which they can replicate is dependent to a large extent on how stable as well as how active the different macromolecules are at different temperatures. So proteins, if you get them too cold, they stop moving around much and they don't work very well. You get them too hot and they denature, they fall apart. And that will define the range of temperatures over which a protein is active. And organism is simply a collection of its proteins and other molecules. And somewhere within that activity of all those different molecules will define where the organism itself can be active. And so we have organisms that are able to be active at very cold temperatures, others that are able to be active at very high temperatures, but every organism is going to have a range over which it's able to replicate and otherwise metabolize of temperatures. And somewhere in the midst of that range, there will be an optimal temperature. And all of that's going to be defined by how functional its different molecules are at different temperatures. And then you can vary things like the pH, and you can vary things like the salt concentrations, and all of these. There's going to be some range over which the organism is functional, and that range is going to be dependent upon at what level of temperature, level of salt, pH, its different constituent molecules are functional. So that's one way of thinking about the chemistry of microorganisms. The other way is what we'll be doing in the laboratory. And that is that in the laboratory we'll be growing microorganisms. And one of the things we'll be doing is looking at the quote-unquote biochemistry of these organisms. And the way we're going to do that is by growing these organisms in the presence of different types of chemicals that are able to, in some way, manipulate either the organism's growth or what the medium the organism is growing on looks like. And the media is going to vary in the types of molecules that we supply to the organism as food, as well as the way that we buffer the pH of the media, as well as the temperature of the media. And so, for example, we're going to have carbon sources. Carbon sources are where the organisms are going to get their basic nutrients, that is their energy and the building blocks, the carbon building blocks of their own cells. in addition to carbon sources, and very similar to carbon sources, are energy sources. Typically they're the same molecules as the carbon sources, but where the organism gets its energy in order to replicate and survive, repair itself. And then we also have sources of nitrogen, and for the organisms we're working with by and large, these sources are going to be either amino acids, or they're going to be combinations of amino acids that make up what we call peptides. Or they could be proteins themselves. In fact, we'll be looking in some organisms at their ability to break down proteins. The gelatinase test is an assay for ability to break down proteins, for example. Then we also have tests where we're looking for the ability of the organisms to break down starches. We could also have a test, although we're not going to do it, of the ability of the organisms to break down lipids or nucleic acids. So these are all things that you're going to be interfacing with in the laboratory. And generally, if something is made of a protein, it's going to be a source of protein / amino acids for these organisms. Just like when we eat proteins, we get our essential amino acids from eating those proteins. So do the organisms. And so it's going to be important in the laboratory to be conscious of the types of chemicals that are found in the media that we're working with. There is a chapter called The Growth of Microorganisms, or something like that. I can find the table of contents quickly. It is Microbial Growth, chapter 6. So in that chapter, they'll be talking more about these different constituents of the medium. But ultimately, it comes down to aspects of chemistry and biochemistry, which is covered in chapter 2. Also, chapter 5 talks about microbial metabolism. And in there, we'll be talking about things like optimal temperature ranges and optimal pH ranges and things like that. So chapter 2 is really just an attempt to get us started in thinking about chemistry, but ultimately our goal isn't so much to understand chemistry as to understand how organisms function in light of these various chemical principles.